Today, we're really going to tie together a lot of the topics that we've talked about in the past week to kind of get an overarching understanding of how we go from our DNA to actually having our traits. How do we actually turn the genetic code that we have into the physical characteristics that we see, like eye color, hair color, height, those different traits that we talked about getting passed on. So we've already talked about a lot of this, but now we're going to fill in these missing pieces of how we kind of go through each step. In a quick review, we've already talked about a lot about genetics, how we get these traits passing from our parents to offspring. But it's not as simple as just having DNA and then having traits. Like we said, there's a series of steps that we call the central dogma of genetics. I briefly talked about this last class, how we go from DNA to our RNA to our proteins. So let's just kind of step back a little bit and have a quick review of our DNA, how that structure works, because it's going to help us understand everything about how we go from having our DNA to having our traits. And so you probably remember a lot of this, that we have our DNA as a double helix. It's these two strands that are made up of these nucleotides, phosphate, sugar, base, phosphate, sugar, base. What's important to remember, these bases here come in 4 different flavors, right? We said they can either come in the form of guanine, cytosine, adenine, or thymine. And these 4 letters here are going to be the basis of our genetic code that give us all of our traits. So when you look at a strand of DNA, you're going to find this genetic code, these letters, kind of in what we would consider somewhat a random order, C, G, A, T, C, so forth and so on. Millions and billions of these letters. But these letters are going to result in all the different traits. You can think of it almost as kind of like the alphabet, but it only has 4 letters. So how do we go from having a DNA strand that just has kind of this random string of 4 letters, G, C's, A's, and T's, to having our traits? Well, there's going to be kind of 2 steps in this process. So the first step is going to be to turn our DNA into RNA. We're going to go through a process called transcription. So if you remember, I said DNA never leaves the nucleus. We turn DNA into a blueprint, a copy of it. Transcription is the process of how do we make that copy. And for us, it's actually really easy to understand how we go about taking this DNA and turning it into a molecule of messenger RNA, a copy of DNA, because it's exactly the same as DNA replication. So we talked about how DNA makes copies of itself for new steps. The process of making a strand of RNA is pretty much exactly the same. The first step is to split open the double-stranded DNA. So we're going to use the same enzyme we used earlier, helicase. It's going to unwind the molecule of DNA. It's going to split up the 2 strands of DNA. The second step is that there's going to be an enzyme called polymerase again, this time called RNA polymerase, which is going to copy 1 strand of the DNA. So if you remember, in replication, we copied both strands, but remember that RNA is single-stranded, so we're going to just copy 1 strand of the DNA. And we're going to do that through our typical base pairing rules. So you can see here, based on my DNA that I've unwinded, I'm building a new strand, this time of RNA, following the base pairing rules. So T goes with A, C goes with G, and the only difference is RNA, if you remember, doesn't have any thymine, doesn't have any T's, it has U's. So you can say, okay, A with U, T with A, G with C, A with U, T with A, et cetera, et cetera, et cetera. So you can see how these kind of mirror image of each other. And like I said, we're only copying 1 strand. So we always call that strand the template strand. There's an uncopied strand, which we just call the coding strand. Call it the coding strand because if you look, it actually has the same code as an RNA. G's here, C's there. Well, there's a U here instead of a T, but it's basically a mirror image of this, or a copy of this strand, just with that one change. So we have a template strand and we're coding strand. And then what's very important to note is that when we make our RNA, we don't copy the entire DNA molecule. We only copy pieces of it. So we copy small sections of it. We copy from a start to an end site, a site called a promoter and a terminator. We just copy a small piece of it. So it's just important to note that we don't copy the whole DNA strand. That would be too much. We just copy the portion that codes for a specific protein we might be building. So maybe for a specific trait or a specific part of our cell. We're just copying 1 piece of DNA at a time. And then what happens is that mRNA just breaks away from the DNA, and then that's ready to go out and make a protein or go out to the rest of our cell. So that's how we go and make our mRNA. Now there's one other thing that I want to mention about mRNA. When we copy our DNA, it turns out that there's a lot of genetic code that we don't use. There's lots of random sections of our DNA that don't even code for anything. We call them introns. Some people have very big introns, some people have very small introns, but basically it's genetic code that doesn't do anything. We don't even fully understand why it's there. Because this genetic code doesn't do anything, it actually gets cut out of our RNA sequence. So you can see here we keep certain sections called exons, and we cut and paste those together. We get rid of all the introns, the pieces of DNA that don't actually do anything. And again, we don't know why we have these introns, these sections of DNA. It's kind of a mystery to science. They're just there, so they get cut out. So once those sections get cut out, we're going to move to our next step, which is translation, which is actually turning that RNA, that DNA blueprint, into a working protein. So here we go from an RNA molecule. Now we want to translate that into a protein. So this process of translation is going to turn our RNA into a protein. The first step of that is that the RNA is going to leave the nucleus. So remember that our DNA always stays safe inside the nucleus. So RNA gets copied inside the nucleus. It's going to travel out into the rest of our cell. And it's going to find that protein building site that we call the ribosome. Remember, the ribosome itself is called rRNA. It's made of RNA itself. But it's going to go ahead and find that organelle, the ribosome. That's kind of like our construction site. That's going to help us build the protein. So here you can see down here I have the strand of RNA that's coming in and going through the ribosome. Now this is where the magic kind of happens. We said these molecules called tRNA bring these things called amino acids. Amino acids are going to build our protein. So tRNA molecules are going to come one at a time, and they're going to carry these amino acids to the ribosome to end up putting the protein together. Now, what you'll notice about the RNA here is that the RNA is going to match up to the tRNA, and it does this 3 bases at a time. So if you actually look here, I have a few tRNA molecules, transfer RNA molecules, and you can see down here, the RNA has this genetic code that got copied from the DNA. The tRNA is going to match up to it. You can see here that AUG is going to pair with UAC, the complementary bases, and that's going to match up perfectly. So this tRNA molecule is going to bring a very specific amino acid when it sees this code. And then here you see UUC. tRNA is going to match up AAG, bring a different amino acid. And AAA is going to match up with UUE. So a tRNA molecule is going to bring a different amino acid. So it's going to be read 3 bases at a time. Our RNA, let me say that again, is read 3 bases at a time. It's kind of like if we only spoke in words that were 3 letters wrong. The cat ate the hat. Sentences that only have 3 letters. Our DNA is consistently and constantly read 3 letters at a time. We call every 3 letters a codon. So codon is like a DNA word, 3 letters long. So you can see here that these RNA molecules are going to pair up 3 letters at a time. We call these bases on the tRNA the anticodon. They're going to pair up 3 letters at a time, and every codon is going to match with a specific amino acid. So anytime you see, like for example, UUU, it's going to match up with lysine, always. So it's always going to be pairing up, and every 3 letters specifically relates to a certain amino acid. And you can see here, this is a great example. Imagine this black line here is our messenger RNA that just got copied from the DNA. It's going to go through the ribosome, and every 3 letters pairs up with 1 amino acid. 3 letters pairs up with 1 amino acid. 3 letters pairs up with 1 amino acid. We're going to keep matching those up. And as it goes through the ribosome, these amino acids are going to chain together. And a chain of an amino acids makes a protein. So once these start to chain together, you're going to have your finished product, a protein. So here we go. The amino acids are going to join together in a chain, and that completed chain is going to make up your protein. 3 letters at a time codes 1 amino acid, and if you keep going every 3 letters and join those together, you're going to end up with a protein. So that's our final step here, the protein. Once we have the protein, that protein is going to give us a different trait. So a different protein from you and I might give you a different hair color. It might give us a different eye color. It's going to code for some different trait inside our body. Now, What we're going to do now, which is pretty interesting, and I'm going to show you how to do this in a second video. I'm just going to give you a quick introduction to it in this video. How do we know which amino acids go with which codons? So what we're going to find out is that since every codon pairs up with a amino acid specifically, we can kind of chart these out and actually read our DNA ourselves, which is what we're going to do. We're going to use a tool called a codon wheel to identify what amino acids go to what codons. And so you can see a codon wheel right here, and you're going to use one of these to decode your RNA. So a codon wheel tells us the 3 letters for each codon in an RNA, and then on the outside it tells us what amino acid it goes to. And you always read it inside to out. So let's say we had the code GCA. Well, that codes for the amino acid alanine. And then if we had GCC, same amino acid, alanine. Then we have AAG, that codes for lysine. So you can see here that, sometimes some codons lead to the same amino acid, but they're always the same. GAG will always give you glutamic acid or whatever. So you see all these amino acids. These are all the different amino acids that can join together to make a different protein. I'm going to point out 2 very important ones right now because you're going to use them in the next activity. So the 2 special codons, the first one is methionine, which is always given by the code AUG. Methionine is important because every protein starts with methionine. So we call it our starting codon. AUG will always be the first codon in any protein. So when you actually go ahead and try to decode your genetic code, you actually have to look for AUG to find that start sequence first. The other thing to note is that there's 2 places on this wheel where we see stop. UGA, UAA, and UAG. These are stop codons, so these mean that the protein stops. If the genetic code were to keep going on and on and on, your protein would get bigger and bigger. So we have specific stop sequences that tell our cells, tell the ribosomes, that there's no amino acid that goes here. This is the end of the protein. So you're going to be using these codon wheels in just a minute to decode your own genetic code. So just a quick overview, and this is one picture that illustrates perfectly the entire process of kind of protein synthesis. Here you have your DNA all twisted up. We unravel it, and through transcription, we turn that into RNA. We read our RNA 3 codons at a time through a process called translation to find matching amino acids that chain together to form a protein. You see here, 1, 2, 3, 1, 2, 3, 1, 2, 3. Those are our codons that tell us which amino acids join together to make that protein. So now we're going to go ahead and you can watch the second video here to actually practice decoding your genetic code and building proteins using that genetic code.